Hey guys, welcome back to Computational Biology. This video disappeared. I had a link on the course page to go to Annotation Part 3, and it doesn't exist anymore. I don't know what I did with it. And I unfortunately lost all my raw files a while back when my solid state drive decided to go to its eternal resting place. So I'm re-recording it. So bear with me because my train of thought from the previous two videos and leading into the fourth video, since this is the third, I'm hoping to recapture it, but it's been a long time since I made these videos. What I have noticed is I have lost some weight since the last time I recorded these videos, which is good. They can be hard to go back and watch again. But so bear with me. I'm sorry that that video disappeared, but I'm re-recording it. Hopefully we'll be able to continue on somewhat seamlessly. So let's go ahead and jump right on into it. Oh, look at that. No, not as big of a turkey neck as my last two videos. Okay, so I believe this is where we left off. So from here, we're going to do from bio seq record, import seq record. The typos are still the same as last time. From bio.seq record, import seq record. All right, there we go. And now we're going to create a seq record. So record equals seq record seq, and I am going to paste this in to avoid typos. So you may want to pause. Why is there two? That shouldn't be like that. And I hopefully have made it large enough. Here, I'm going to move this over this way a little bit. And oh, that's way too big. Oh, all right. So you can pause it here. I made it way too big. I'm going to go back. All right. So we're creating the seq record. And now we are going to print it. And we're going to print it as a FASTA file, or FASTA, whatever you want to say. All right, so now we've created the sequence record, right? So this is how, when we look at FASTA files from NCBI or wherever else you may get them, they're in this format where we have our header, and this is for Cucumis sativus, and it's a chalcone synthase, and here is our amino acid sequence for it. Let's see. Now if we just do, instead of saying what type of format as a FASTA file, we can just do print record. When we do that, you see that we get it in a different format, where instead of having the whole sequence, it kind of breaks it up where we have the ID of the sequence. We have a name, which we have an unknown name, but this, because we didn't denote a name up above, but this, you could put whatever you want in there, really, but if it's human or something like that, or I don't know if it's traditionally that they put the scientific name there or the common name. Like I said, this has been a long time since I recorded the last videos. But then we have the description of the protein itself. You probably could put, so it's the description is probably the name, and then we probably put the scientific name up there if we were to do this over again. And the number of annotated features, we didn't enter any. And then it's the sequence. But you notice with this record that the sequence isn't the full sequence. It gives you the first
however, was it 30 or so probably amino acids, and then the dot 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 means there's a bunch more in between, and then we end with SAC. So as you can see here, it ends with SAC. Okay, so now we already have, so from bio import seq IO, which I think we already have imported, but we'll just do it again because it doesn't hurt. Record equals, and we're going to actually read in a record now that we have downloaded. seqio.read, and do nc 0 5 8 1 6 .gb .txt. We're going to say genbank is the format. And now we're going to just say record to print record. And so as you can see, we have our seq record here with our amino acids. Nucleotides and amino acids. Nucleotides, so this is a DNA sequence. It's a plasmid. We have the complete sequence. A bunch of good information here. We can also ask for the length of the record for this. So we can say, how long is it? It's 9,609 base pairs. We can say, how many features are there? So we'd say length of the record features. 41 features within this record. You can print the first or the 20th feature. So say, what's the 20th feature? It's a gene. Its location is between base pair 4,000, well, including 4,342 to 4,780 on the plus strand. The gene ID, for if you're going to look this up on like NCBI, is 2767712, etc. What else can we look up here? We can also do, what did we do? Print record features. We'll do another one. Print record features, we'll do 21. And as you can see with this one, this feature is a CDS. And so the location for this is 4,342. So this is actually an overlapping, but this is CDS instead of the actual gene. And the location is the same. As you see, we have our some of our keys. We have where our codon start is, the gene, the value. What else do we have in here? Most of the same information. So for this note here, it's similar to many previously sequenced pesticid immunity proteins, entries of Yersinia pestis. So it's got a little bit of information on what the function of this gene is. And also we have this translation value, so we have the actual amino acid sequence for record 21. So we can subdivide the record. So if we take our sub record and we say it's from our record, we're going to say we're going to take this area here, right? So we're going to take 4,300 through 4,800. So that's some upstream and some downstream of this gene. And we can say what's the length of our sub record. And it should be 500, right? 500, perfect. How many features are within this? So remember there's 41 features, did we say? 41 features. So when we subdivide, it preserves how many features and stuff are within that subdivision, or it should. So if we say, what's the length of sub record dot features, it will tell us how many of those 41 features are within that 500 base pairs. And if I could sub underscore record features. 2. So there's 2 features within this area, which makes sense because we had the gene and the CVS that were both features in that area. And let's see. And remember, because Python is funny and it starts with numbering at 0, so if we want to look at the first of those 2 features, we do subrecord. So there's really 40. Let me just take a step back. So there's really 42 features, right? Because we start at counting 0. And so if we do subrecord feature 0, we are locating that gene again. And so if we do subrecord features 1, we should locate that CVS again. Yep, the CVS. We're cruising right along here. So now if I do print sub record features 0, we should pull up that same one that we pulled up before. So it looks essentially the same, except you'll notice, so we have the gene, remember 2767712. If we go up here, we have the gene 2767712, so that all stays the same. All this information stays the same, but the location changes because we're looking at the subrecord, right? So no longer are we looking at the location in the context of the entire plasmid. Instead, we're looking at the sublocation in the context of our subrecord, or our location in the context of our subrecord. So it should be the same too if we do print subrecord features, and then we do 1, right, where the CVS is. So all that information except the location should stay the same, which it does, right? So we still have sequenced pesticin immunity protein series of Yersinia pestis. The translation remains the same because that's all preserved even when we do these subrecords. The only thing that's different is the location is now 42 to 480 instead of 4,342 to 4,780. Okay. So pretty cool, being able to manipulate it without like losing a lot of this information. Okay, we can also do sub-record annotations, and if we look at the annotations, we say that we have the annotation for the molecule type is DNA, which makes sense because it's codons, it's not a protein sequence. Sub record dbx refs. Nothing. Okay. Sub. I don't know why I put that in there then. Record annotations. Let's look at topology. Equals linear. Now, okay. So when we do this, we can add to our annotations of that record, right? So if you wanted to add that this is from a certain sample or something, an iteration or a trial or something that you're doing, you can kind of add notes or annotations to your subrecord. So now if we do subrecord annotations, we print that again, you can see that instead of just saying molecular type DNA, now we have molecular types DNA and the topology equals linear. So topology, that's meaning like a plasmid would be circular DNA, and since we just have a chunk, we could say that it's a linear piece of DNA. What else can we do So we can look at the ID, subrecord ID, which was in all that information, but we should have record. This is our NCBI identifier. Somewhere it should be in here. NC 05816. Where did that come from, I wonder? It's gotta be in here. You've probably seen it in the same. It's right there. Oh, no. That's the name of the file that we loaded, right? Yeah. Okay, so that's the name of the file. So perfect. So the ID and the name sub in this case are the same, well, similar. So the name is going to be without the .1. Let's have a description of this. Description. So this is the Yersinia pestis biovir microtus. So it's probably biovariant microtus strain 91,001. The plasmid is PCP1, and it's the complete sequence. Although that's not correct for us, right? Wait a second. Okay, so we're going to change this. Remember, because subrecord description, we just need to take and copy this. We cut a big chunk out, right? So this isn't complete sequence. So we're gonna make this now say partial for this one. So this is showing that we can edit these files. So partial sequence is what we want it to say. So now if we take this and just run it again, it should say partial sequence, which is great because we don't want to mislead people and think that this perfect 500 base pair snippet is the entire sequence of this Yersinia pestis. Okay, so all right, one more thing. Print sub record format genbank. We're going to say everything through 200 and ... Okay, so we have the GenBank record now for this, where we have, it shows the locus, how long it is, what type of molecule, DNA versus amino acid, or protein sequence, that it's a linear piece. It was first sequenced in 1980, I guess, or the sample was collected in 1980. I don't know. I don't remember what that is from. Anyways, so the definition is what our stuff is, and you can see we edited what our sequence is. We edited it so it says partial sequence, which is great, and we have the accession there, and that is it. So I hope
this fit. Whoa, that was weird. I hope this fits a bit more smooth, or fits smoothly into the remix. I was thinking about at the beginning of the video saying that this was a remix, and then getting like the horn from like you know, the club. But then I was like, man, I would be a loser if I did that. But I just told you the joke, and so I still am kind of a loser for dad jokes. I'm a dad. I can make those jokes. So hey. All right. This was actually kind of refreshing. I haven't recorded a video in a while. I'd like to get some stuff up, but you know, grants and other duties of being a professor. So that's the end of this video. Thank you for watching. I'm sorry that this video went missing. I don't know why it went 1, 2 to 4. I did record it, and I have a link to a video that doesn't exist anymore. But thanks for watching. I hope this was informative. If you're watching on YouTube, please hit like, please hit subscribe. I will be posting more videos in the near future. I think we're going to get into some biological modeling and whatnot. But thanks for watching, and I will catch you in the next video.